Good afternoon, Dr. Wan, Savani, and Norita. We are from Group 7. I'm the group leader. My name is Lee Zhongzhe. My metric number is 23005109.
My name is Muhammad Haziq bin Fazanwa, and my matrix number is 22002069.
My name is Lin Hui Chi, and my matrix number is 22121782.
Good afternoon, my name is Ding Yabo. Matrix number is 221148676.
Hello, my name is Farafazana binti Azharsham, and my matrix number is 22001096.
Contagion Movie is a 2011 thriller that depicts the global spread of the deadly virus MEV1 that originated from bat and pig in Hong Kong. The narrative unfolds through multiple perspectives, showcasing the responses and struggles of governments, medical experts, and the public in the face of a well-spread pandemic. The storyline portrays the challenges and consequences of a global health crisis. RNA viruses such as influenza virus, COVID-19, and Ebola are leading to substantial morbidity and health burden. The variability in host responses will affect the dynamics of the innate immune response, which will result in chronic disease. In Contagion Movie, scientists use genomic sequencing to analyze the genetic makeup of NEV1 virus and track its origin in order to gain insight into its behavior, transmission pattern, and potential mutation, and thus develop a vaccine.
Today, we are going to present about genome sequencing. So, let's dive into genetics to understand how scientists decode this life blueprint to genome sequencing. So, our first problem is, what is genome sequencing? As we know, every living things, including bacteria, plants, and mammals, all of them have a distinct genome of nucleotide bases which are including adenine, thymine, cytosine, and guanine. An organism's distinct characteristics like their fingerprint or their pattern. All of this can be determined by us if we know the bases in that organism. Thus, determining the order of these bases is called sequencing. The whole genome sequencing is a laboratory procedure that can help scientists to determine the order of bases in the genome of an organism in one process. Our second question is, what are the purpose of genome sequencing? Is it useful in our life? The answer is totally yes. Genome sequencing plays an important role in protecting humans' health. For instance, genome sequencing is an effective tool in environmental monitoring, pathogenic design and identification. And also, genome sequencing helps scientists to individualize vaccine. And I will now introduce some important events in the history of genome sequencing. The first important event is in 1977, which the Fenwick Singer have developed the first DNA sequencing method, and this method is called chain termination method. The next important event is in 2005, where Jonathan Robert and his colleague have successfully developed the first next generation or second generation sequencing method, the 454 system. But in nowadays, the main player of second generation sequencing is Illumina. And also, in 2010, the PacBio have developed the first third generation sequencing method, which is single molecule sequencing in real time, SMRT. And also, in 2012, the Oxford nanopore technology have successfully developed the fourth generation sequencing, which is nanopore sequencing. The fourth generation sequencing is more popular than third generation sequencing, as the fourth generation sequencing brings a more outstanding benefits than third generation sequencing, and both of them have the similar method.
Now I will explain about the procedure of genome sequencing. So the first step is to obtain a sample collection. So there are three ways to obtain sample collection, which is through saliva, tissue, and even blood. So from saliva, there are two ways of swab and saliva collection tube, and tissue is through skin biopsy, which is the extraction of tissue from the skin, and for blood is through vein puncture, which is the normal way to obtain a blood sample. The second step is DNA extraction. This step is to purify DNA from other cellular components, and it involves lysing the cells and solubilizing the DNA. And the methods are through chemical or enzymatic methods, and some of the methods are called phenolchlorophore method, sorting out and protein escape treatment, and silica gel membrane. So the third step is DNA shearing, one of the most important steps. So this step involves cutting DNA into ideal pieces to be read by the PCR machine. So in order to cut the DNA are through molecular scissors, which is enzyme or even mechanical disruption. The fourth step is DNA library preparation. This involves duplicating the DNA fragments to amplify the genetic material, and this will help the detection and identification of gene sequence. So from the DNA library, we will now proceed to DNA sequencing. So this is to determine the order of nucleotides and DNA strands. So most of sequencing is done by the next generation sequencing technologies. So those DNAs are now called DNA reads. And after we obtain the DNA reads, we can now analyze the DNA using computer programs or even other methods.
One of the platform of genetic sequencing is Illumina sequencing. The first step is library preparation. Next generation sequencing library is prepared by fermenting a gDNA sample and ligating specialized adapter to both fragment ends. The next step is cluster amplification. Each brown fragment is amplified into a clonal cluster through bridge amplification. The third step is sequencing. This step is repeated n times. Sequencing reagent are added and the first base is incorporated. The flow cell is imaged and the emission from each cluster is recorded. The emission wavelength and its intensity are used to identify the base. The fourth step is alignment and data analysis. Reads are aligned to a reference sequence with bioinformatics software. The development of Illumina sequencer, it is a renowned producer in the diverse sequencing industry. First, they implemented a sequencing biosynthesis strategy that uses fluorescently tagged reversible terminator nucleotides on clonally amplified DNA scaffold that are immobilized to an acrylamide coating on a glass roll cell surface. Illumina provides a diverse array of sequencing platforms categorized into benchtop sequencer and production-scale sequencer. Among production-scale sequencers, NovaSeq 6000 exclusively supports the whole genomic sequencing of humans, plants, and animals, offering functionalities akin to benchtop sequencer. One of the strengths of Illumina sequencer is ensure 99.9% accuracy in sequencing. The incorporations of a blocking group allow the efficient sequencing of homopolymeric regions, as only one nucleotide is added per cycle. According to research done by Quill and other researchers, MySec, which target the market for clinical diagnostic and smaller laboratories, only has 0% of some error-free rates. The next trend is deliver high-throughput data swiftly, as only require a minimal sample amount per run. Limitations of Illumina sequencer is lack of synchronizations in the synthesis reactions across individual members of the cluster. This can disrupt the accurate generations of a consensus sequence and impose constraints on the number of cycles that can be executed. Other limitations is overclustering. Another platform of genetic sequencing is nanopore sequencing. After sample collection and library construction, nanopore sequencing will proceed by passing the DNA molecule through the nanopore. Each nanopore corresponds to its own nucleotide connected to the channel and the sensor chip, which measure the electric current that flows through the nanopore. When DNA molecule passes through the nanopore, the current is disrupted to produce a characteristic squiggle that decoded using base calling algorithm to determine the DNA and RNA sequence in real time. The development of nanopore sequencer, it is exemplified by the innovative technology pioneered by Oxford Nanopore Technologies, which directly detect nucleotide as long stretches of single-stranded DNA passes through a protein nanopore embedded in an electrically resistant polymer membrane, for example nanopore minion. It enables direct sequencing without the need for DNA synthesis or amplifications, thus demonstrating efficiencies in disease diagnosis at a low cost. It has the advantages of whole genome sequencing and disease diagnosis with fast speed and cost-effective performance. One of the strengths of nanopore sequencers is capacity to sequence DNA of virtually limitless length, with demonstrated sequence rate exceeding a megabase. Another strength is lack of image analysis step, thus facilitating rapid detections of target DNA in clinical samples for pathogen screening. Last, it is highly portable as it has compact design and powered via USB. One of the limitations of nanopore sequencers is relatively higher error rate compared to short-read sequencing like Illumina sequencers, error encompassing both insertion and deletions, hence may require additional short-read sequence data for systematic error corrections. Next, data yield variation poses challenges in predictability.
One of the limitations of genomic sequencing is its read length and accuracy. Some genomic sequencers have limitation in terms of read length and accuracy. This limitation affects the ability of genomic sequencing to assemble and interpret certain genomic regions accurately, hence causing difficulties in identifying some types of genetic variants such as dementia and Huntington's disease. As an example, a well-known second-generation genome sequencer, Illumina, only has a read length between 75 to 250 base pair. To improve this limitation, some efforts were made. One of the ways is to use long-read sequencing. A biotech company called PacBio introduced a third-generation genomic sequencer known as SMRT, which has a read length higher than 1 kilobase pair. SMRT also has greater certainty in assembling and interpreting repetitive DNA regions. SMRT is very helpful in targeted sequencing, full-length mRNA sequencing, and more complex population sequencing. Read length and accuracy issues are also overcome by sequencing technology that produces long read output while using short read sequencer. This technology is introduced by 10x Genomics. They combine the traditional Illumina sequencing method and synthetic link read technology to produce long read output. The second limitation of genomic sequencing is the limited amount of viral genome when it is used for viral sequencing. This limitation occurs as viral genetic materials often come with low number compared to the DNA of a host. Moreover, several viral variants can coexist in a single sample, causing more or less variable sequences to present. To combat this limitation, a complete sequencing and analyzing protocol called VASAP was introduced. This protocol enables scientists to generate dominant viral genome sequences rapidly from samples. It is first evaluated using OC43 coronaviruses, and the result is quite satisfactory. The protocol managed to obtain a complete sequence viral genome for 7 out of 11 clinical samples.
Genome sequencing, while having existed for hundreds of years, was historically fraught with significant limitations and drawbacks. However, advancements in technology have allowed for the development of workness and more sophisticated applications of the process. And now, with the development of science and technology, genome sequencing can help people find disease more effectively and accurately.